Hi everyone, and welcome to our AlcoR tutorial. In this tutorial, we'll discuss the AlcoR tool and its usage in the analysis and visualization of low complexity regions in FASTA data. Let's start by understanding what are low complexity regions and why they are relevant in bioinformatics. So, what are low complexity regions? Low complexity regions, or LCRs, are areas in sequences of elements that contain low complexity or repetitive elements. These can include tandem repeats, inverted repeats, homopolymer tails, GC-biased regions, similar genes, hairpins, and more. In bioinformatics, it's crucial to identify these regions because of their association with regulatory and structural characteristics. This information can provide valuable insight into gene expression, genome stability, and other important biological processes. However, identifying LCRs can be challenging because of their high coverage depth, ambiguous read mapping, and other factors. This is where AlcoR comes in. It provides a solution to these challenges by automating the process of LCR identification and quantification. AlcoR works by generating parallel bidirectional complexity profiles of the input sequence. The first step involves compressing the sequence in two directions, from the beginning to the end of the sequence, left-right, and from the end to the beginning of the sequence, right-left. Then, the method takes the minimum of both directions, performs smoothing, and segments the regions that fall below a certain threshold. The final step involves visualizing the identified regions by generating a map. The bidirectional computation is critical to the AlcoR method, as it ensures that even original subsequences that are copies of other sequences are identified as low complexity regions. By processing the two directions in parallel, the AlcoR method reduces computational resources substantially. The tool consists of 5 modules: information, extraction, mapping, simulation, and visualization. These modules work together to provide a complete solution for low complexity region, LCR, identification and quantification. The information module retrieves information on the length and GC percentage of each FASTA read. This information can be useful in later stages of the analysis. The extraction module allows users to extract a sequence from a FASTA file using positional coordinates independent of the existing headers of the FASTA files. The mapping module is the heart of the AlcoR method. It computes the low complexity regions in a FASTA file, providing bidirectional complexity profiles and structural similarity analysis. This module utilizes the parallel bidirectional complexity profiles discussed previously to identify LCRs accurately. The simulation module performs FASTA sequence simulation with features such as file extraction, random generation, and sequence modeling, with SNP-specific mutations. This module allows users to test the accuracy and robustness of the AlcoR method. Finally, the visualization module creates an SVG file with a map containing the low complexity regions. This map provides a clear and visually appealing representation of the LCRs identified in the FASTA file. Let's have a look at how to use AlcoR in practice. To install this tool, you can use Conda installation or clone the repository directly from GitHub and compile the source code. After installation, we can now use AlcoR freely. To view the information on the usage of each module, use the help command like this. The output is information regarding what each submodule does. To view submodule specific information and parameters to use, write AlcoR command and help. It outputs possible parameters that can be used. Now, let's see a couple of examples. First, let's make a demonstration which shows how to use the AlcoR to analyze a simulated FASTA sequence. The first step is to create a synthetic sequence using the simulation command shown on the screen. This command generates a FASTA file named sample.a.fasta. As you can see, this is a FASTA file containing a DNA sequence. Next, using this sequence, we will run the AlcoR mapper 3 times with different thresholds: 0.5, 1.0, and 1.5. The goal of using different thresholds is to showcase the impact of threshold selection has in the identification of LCRs. These commands produce the 3 positional files, respectively. After this, we run a function that converts the positional data in the txt files into a profile that can be plotted. Finally, we use GNUplot to generate a PDF named AuDA to showcase the complexity map of the FASTA sequence for different thresholds. As can be seen by increasing the threshold, AlcoR can better detect LCRs with more mutations in this simulated FASTA. In this second demo, we will be going over the process of using the AlcoR tool to map low complexity regions in a set of viral genomes. After downloading the viral genomes, the first step is to set a threshold value for the low complexity regions, which in this case is set to 1.2. Next, we have a list of 8 herpes viridae viral genomes for which we will map low complexity regions. These genomes are HSV1, HSV2, VZV, EBV, HCMV, HHV6A, HHV6B, HHV7, and KSHV. For each of these genomes, we will be using the AlcoR Mapper module to map the low complexity regions. We will run the Mapper module twice for each genome, one to get the distant low complexity regions and another to get the local low complexity regions. Finally, we will use the AlcoR visualization module to visualize the low complexity regions for all 8 genomes in a single map. The options used for this module are omap.svg to specify the output file name. Other parameters, such as colors used and line width, can also be specified. And that's it. As we can see in the generated image, using these steps, you easily create a map of local and distant low complexity regions in a set of natural viral genomes using the AlcoR tool. In addition to mapping LCRs in viral genomes, AlcoR can be applied to large genomic data since the tool's mapping and visualization capabilities are scalable and can handle large amounts of data. An example of the Homo sapiens genome with identified LRCs is shown here. And here is another example using the cassava genome. Using AlcoR for mapping LCRs in large genomic data provides several benefits, including automation of the process, reduction of computational resources, and visualization of the results in a clear and concise manner. Furthermore, since the method is based in data compression, the complexity profiles do not underestimate while still having high sensitivity. This can help researchers quickly identify and quantify LCRs in their data, saving time and increasing efficiency in their work. Thank you for joining us for this tutorial on AlcoR. Feel free to email us